Good. Okay. All right. So it is just about the hour. So I'll go ahead and introduce our speaker today. It is my great pleasure and honor to introduce our outside speaker today. Dr. James DeLany is a senior investigator at the Advent Health Orlando Translational Research Institute. He began his independent research career in 1989 at the Pennington Biomedical Research Center, where he was a faculty member engaged in the study of energy and substrate metabolism for 16 years. He expanded his emphasis on diabetes research after being recruited to join the faculty at the University of Pittsburgh in 2005. A major focus of his work in 2018 is an NIH-funded project applying PET-CT imaging to determine the role of specific steps of glucose uptake and the contribution of muscle and adipose tissue in the lower peripheral glucose uptake in African-American women. He is also examining the role of mitochondrial function and racial differences in metabolic health, aerobic fitness, and glucose uptake. In another NIH-funded project, he is employing doubly labeled water, indirect calorimetry, activity monitors, and dual energy X-ray absorptometry, all of these familiar things that we do here in our studies to examine the role of energy balance in the increased obesity associated with the recently identified CREBRF missense variant in Samoans. So a little bit of history on Dr. DeLany is that he received his PhD from The Ohio State University back in 1986. He's published well over 100 peer-reviewed research articles, written many book chapters, and his work, of course, has been funded by the NIH, what seems like several decades, many decades. So we are certainly lucky to have Dr. DeLany here today. He has agreed to take time out of his busy schedule to fly back and forth and back home again to try to visit us here in Columbia, but he will share his work through the Zoom world that we have become very familiar with. Anyway, so he's going to share some recent work on a topic that is near and dear to many of us, which is mitochondrial function and insulin sensitivity. And I think he's going to tell us about how this affects African-American women in particular. I, for one, am very much looking forward to his talk, and I'm sure you are too. So without further chatter, please join me in welcoming today's speaker.
Well, thank you for the nice introduction. So I'm not going to talk much about the PET study because we really don't have much data to talk about, or the Samoa study, which I'm still blinded to the genotype. So we're still collecting data in Samoa, but I'm going to talk about some other data, the mitochondrial functions in particular. So the reason I got involved initially in the study of African-American women racial disparities was because the prevalence of obesity is over twofold greater in African-American women compared to Caucasian women. Whereas for men, at least when I got started in this area, the odds ratio there wasn't a significant difference in African-American women, but now in the African-American men. But in the latest report, at least in this 2008 report, the odds ratio for men is now significantly greater, but it's only 1.13. Furthermore, when I first started this area, the prevalence of diabetes was over twofold greater. It was like 2.4-fold greater in African-American women compared to Caucasian. It's now reduced some to 1.9, but it wasn't because diabetes prevalence was lower in African-Americans, it's because of the increase in the whites. And in men, there is a racial disparity in men, but it's only about 50% greater instead of twofold greater. And then I wanted to point this out, the Hispanic, I haven't focused on Hispanic until recently, and the prevalence is 1.7-fold greater in Hispanic women. So that's how I got into this area, or why I got into this area. So the reasons for the increased diabetes in African-American women are not completely understood. Lower insulin sensitivity has been reported many times in African-American women. But visceral and hepatic lipid accumulation, which are strongly associated with insulin resistance, are lower in African American. but in limited data suggests lower fat oxidation and oxidative capacity in skeletal muscle of African Americans. So here's just sort of a summary of a lot of the data showing lower peripheral insulin sensitivity in African American women in lean, overweight, and obese. And when I look at this data, it looks like the prevalence, the racial disparity increases with increasing obesity from lean to overweight to obese. And there are these two studies for Barbara Gower's group showing no difference in obese African American women. Well, there were some men in this study, but she has a very high insulin dose in her clamps. And I'll discuss that again later. And when I started in this area, there was no data on hepatic insulin sensitivity. Now that's our study. But after I got started, Barbara Gower's group had reported using an IVGTT in a stable isotope tracer that both peripheral and liver insulin sensitivity were lower in African American women. So one thing that I needed to point out, and I haven't really appreciated, I didn't appreciate initially, and I've never followed up on this, is because we don't do IVGTTs. And to get acute insulin response to glucose, you need to do an IVGTT. So you can see here, this is a meta-analysis of a lot of studies in Africans, African descent in the open squares, Caucasians in the gray squares, and East Asians in the dark circles. And you can see that insulin sensitivity is lower in the African Americans, but they maintain a normal disposition index by increasing their insulin secretion. And so that they can maintain normal glucose tolerance at a low insulin sensitivity by increasing insulin secretion. But once they're no longer able to maintain the insulin secretion, leads to impaired glucose tolerance and then eventually type 2 diabetes. So there's been several studies showing lower visceral lipid accumulation. We found that even individuals with severe obesity, African Americans, had lower visceral fat than Caucasian women, and there was a trend for increased HOMA for the same amount of visceral fat. And we found the same relationship for liver fat. Now this is by CT scan, so the scale's a little wonky. A lower ratio of liver to spleen indicates higher adipose tissue in adipose liver fat. And again, we found lower liver fat in African Americans, but a trend for higher HOMA for the same amount of liver fat. So that sort of serves as a backdrop for the first study. We wanted to examine the mechanisms for the decreased insulin sensitivity. Can you see my pointer on the screen?
You can.
Okay,
yes.
Good. Okay, I know sometimes these things don't work right, but good. And so our hypothesis was that low type 1 fibers in skeletal muscle, reduced mitochondrial content and oxidative capacity led to decreased fat oxidation, which would lead to triglyceride and lipid accumulation, metabolite accumulation, and that would lead to decreased insulin sensitivity. So it was sort of an involved protocol for the participants. They came in for a visit for physical activity monitors, DEXA VO2 max, and then a submaximal exercise test with 1,2-C13 acetate, so that we could assess individual acetate correction factors for the palmitate submaximal exercise test. And then that wrapped up with an overnight stay, standard meal, biopsy, and then the hyperinsulinemic glycemic plan. So we enrolled 22 Caucasian and 22 African American and 22 Caucasian women matched for age, BMI, and weight. And when we do that, we generally see higher fat-free mass in the African American and lower fat mass. And in this case, it was actually a lower percent body fat in the African American women. Also, waist circumference and trunk fat by DEXA, two surrogate markers of central adiposity or visceral fat, were also lower in the African American women. So since physical activity is important for a lot of these things that we're measuring, we assessed physical activity with the body media armbands, and we found no difference in moderate activity, vigorous activity, or steps per day. But we did find a significantly lower VO2 max, which is usually observed in African-American women, about 20% lower VO2 max. Now, you may have not have heard of this body media armbands, and if you hadn't, you're never going to again, because Jawbone bought out Body Media, and after a year or two discontinued the devices, and they just wanted Body Media's patents to sue Fitbit. We're very disappointed. Those of us that have used these devices are very upset about it, because you can see here a plot of total daily energy expenditure by armband versus total daily energy expenditure by doubly labeled water. And the relationship is remarkable for activity monitors. I mean, with an activity monitor, you're lucky to see an R of 0.3 between these. And so I had never heard of them before I went to Pitt. John Jakizik and Brett Goodpasser were going on about these devices, and I saw how good they were. And now we still have a few left. I'm still using them we're still using them in a few small studies, but I've had to switch to active graphs, I guess. Not happy about that. So our insulin sensitivity protocol was a standard two-step hyperinsulinemic glycemic clamp. We used 15 milliunits per meter squared to assess hepatic insulin sensitivity, and 40 milliunits per meter squared to assess peripheral insulin sensitivity. And we conducted indirect calorimetry to assess metabolic flexibility in response to insulin infusion. So we found significantly lower peripheral insulin sensitivity, even at early time points of the low dose, at steady state of the low dose, and at steady state during the high dose insulin infusion. But we found no difference in hepatic insulin sensitivity, assessed by suppression of endogenous glucose production. And our primary measure was the suppression at steady state during the first phase of the insulin clamp. If anything, the suppression was a little greater in the African Americans, but that could be due to they had higher insulin levels as well. So in summary, the lower insulin sensitivity in lean young African American women was specific to skeletal muscle and not liver. This deviates from the typical observed pattern of affecting both tissues, and physical activity levels were similar in African American and Caucasian women. So therefore, findings differences cannot be due to differences in physical activity. So our skeletal muscle characteristics, we did find the expected lower type 1 fibers in African American women. It's not remarkable, but it was significant, and higher type 2. However, we did not find increased lipid accumulation, at least as assessed by oil red O, in either type 1 or type 2 muscle fibers. We did find lower oxidative capacity as assessed by SDH, and we did find lower mitochondrial content by electron microscopy. And we found lower mitochondrial respiration at state 4 of basal, and every step of the ATP titration for estimation of state 3 respiration, and during state U or maximal uncoupled respiration. They were significant even when adjusting for the lower mitochondrial content. So we next wanted to look at the relationship between metabolic characteristics that are usually associated with insulin sensitivity. And we found no relationship, probably because it was a homogeneous group, but body fat wasn't associated, VO2 max, physical activity wasn't related to insulin sensitivity. Type 1 and type 2 fibers, which were different, and it was not even close to being significant. The only one that came close was the oil red O, tended to be inversely associated with insulin sensitivity. Mitochondrial content, which was significantly lower in African American women, was not associated with insulin sensitivity. The only thing we found was mitochondrial respiration was associated with insulin sensitivity. And although this association has been observed sometimes, not always, but this association between mitochondrial content or function and insulin sensitivity, the mechanisms are unclear. But one postulated mechanism is that reduced mitochondrial capacity leads to low fat oxidation and then lipid accumulation. And so we had several measures of fat oxidation. One, which is not very robust, is basal RQ was not different, or steady state 1 or steady state 2, if anything, it was actually higher increase in RQ in response to insulin. So increased carbohydrate oxidation in response to insulin. We had anticipated that they would be metabolically inflexible and not respond as well, but they did. And so that was different from our hypothesis. So we looked at total fat oxidation during a submaximal exercise test, and unadjusted, it was a marginal racial difference. However, if you may recall the lower VO2 max, the 20% lower VO2 max, these individuals, the African Americans, then had a lower exercise intensity because we did 50% of VO2 max as the level of the exercise, and so they had a lower watt. They were expending less energy. They had a lower physical activity, lower energy expenditure. So when we adjust for the lower exercise intensity, there was no difference. Another way to normalize without doing any statistics is to look at the percent of calories that came from fat during the exercise bout. And there was also no racial difference at any time point between the African Americans and Caucasians. So for the tracer studies, we had to do 2 submax exercise tests, one for the acetate correction and then one for the palmitate infusion. So I wanted to look at how repeatable these VO2s for these submax tests were. And you can see here an extremely good relationship between the first test and the second test, and nearly identical mean values for the groups. The other thing I was concerned about is that we had a wide range of watts that the subjects exercised at, from about 30 to 90, and the relationship between VO2 and watts was also remarkably well. So I wasn't too concerned about differences between the 2 tests. And then here's just a little calculation sheet. I just thought I'd show that up there for Elizabeth. So yeah, it's a lot goes into that. So the acetate correction factor increases as the exercise time increases, and it also increases with exercise intensity. But there was initially a difference in recovery in the African Americans, but when adjusting for the lower exercise intensity, it was nearly identical. So where did, okay, last one. I don't think this is the latest one. Okay, so oh no, it is. So this is the tracer data. So with the tracer data, we could look at plasma, fatty acid oxidation, and IMTG, intramuscular triglyceride fatty acid, that's released within the muscle to fatty acids for oxidation. And so as with total fat oxidation, initially unadjusted, there was a significant race effect, marginal. But when adjusting for the lower energy expenditure, there was no difference in fat oxidation. And there was no difference in IMTG fatty acid oxidation before or after adjusting for the lower exercise intensity. So, I really tortured the data to see if I could find something. And so here I did some regression analysis and tested the racial difference in combined slope and intercept simultaneously, slope alone or intercept alone, and there's no difference in plasma fatty acid oxidation or in IMTG fatty acid oxidation. It just wasn't. In addition, we had some ex vivo fatty acid oxidation in skeletal muscle homogenates, and there was no racial difference in complete fatty acid oxidation, acid soluble, or in the ratio between the two. So, and we had one additional measure. So in the high resolution respirometry procedure, we had one chamber with carbohydrate only, and then we had another one where we had carbohydrate plus palmitoyl carnitine. And I did this to look at fat oxidation. You know, and in both African Americans and Caucasian, there was a significant increase in respiration with the added palmitoyl carnitine, but there was a similar increase in the African American and Caucasian women. So there was no difference with that in fat oxidation there. So fat oxidation, you know, once adjusting for lower exercise intensity, was not lower. And we have 4 lines of evidence that it's not. The only one that was a hint of a difference was during the high insulin infusion during the clamp, where they had a higher RQ or higher carbohydrate oxidation. So the preliminary data that I had originally used to develop this project was 3 early studies and small studies that did show a racial difference. But here was a study that came out later that shows that using RQ during exercise at the same wattage, there's no racial difference in fat oxidation. And in fact, in this group, African American women had a lower RQ or a higher fat oxidation at rest, and at two points, the later time points during recovery. So I'm not the only one to show no difference. So this is the summary. We see we did find lower decreased insulin sensitivity. We found that it was only in skeletal muscle and not liver, like we expected. We did find lower type 1 muscle fiber content, and we found reduced mitochondrial content and oxidative capacity. However, we saw no difference in fat oxidation. We saw no difference in triglyceride accumulation, and we found no metabolic inflexibility. But we did find a relationship between mitochondrial respiration and decreased insulin sensitivity, so sort of focused on mitochondrial function entirely. So one hypothesis about mitochondrial capacity is that it's not mitochondrial function, respiration per se, but it could be a biomarker of differences in mitochondrial metabolism or function that lead to insulin resistance in skeletal muscle. And there's quite a bit of data on differences in fission, fusion, and mitophagy being related to insulin sensitivity, and altered mitochondrial function leading to increased generation of reactive oxygen species and oxidative stress, or other mechanisms. So to begin to look at that, we looked at some proteins involved in the fission, fusion, and mitophagy patterns. We looked at the proteins outlined in red. We looked at some from each of these pathways. And we did find racial differences in the FIS1, mitofussion 2 were lower in African American. We found higher OPA1 and Becklin, and lower LC3B, no difference in DLP1, which is now called DRP. And we found some pretty strong relationships between some of these proteins and insulin sensitivity. Mitofussion 2, FIS1, and LC3 were all positively associated with insulin sensitivity, and Becklin 1 was negatively associated with insulin sensitivity. And we found no relationship between DRP1, but we looked at total, not the active form, so we don't need to go back and look at the active form. So as an exploratory aim, I sort of added these qPCR arrays that were mitochondrial arrays to see if we could get some idea of is there a particular pathway in the mitochondrial that is affected and related to insulin sensitivity. And we found some differences. Some of them are actually quite interesting, but we haven't followed up on this at all. So we did find a set of genes, mitochondrial genes, that had an R of 0.64 that were associated with insulin sensitivity. And then the protein content, mitofussion 2 and Becklin 1, we already saw those earlier. But so we haven't followed up on this yet, but there are some good leads in here about some sort of pathways, but it's a lot of work to figure this sort of thing out. So as a follow-up to this study, we developed what we call the MAT-PET study. It's the PET study. And I was working, Julie Price is the MPI on this project. And when we started this project, while developing this project, we were both at Pitt. When we submitted the revision, she was thinking about leaving. And by the time we got funded, she had gone to Mass General. And I stayed at Pitt. And so when I started this project, well, I never started it, but I got ready to start it. I was at Pitt and she was at Mass General. Now, neither of us are at Pitt. And so, but we did get going. And so I submitted a competitive revision that was successful, and that's to add a Hispanic group, which is supposedly easy to do. In Orlando, we have a large Hispanic community. And to add metabolomics and lipidomics analysis to all of the participants. So the main aim of the study is to look at the steps of glucose delivery, transport, and phosphorylation, and then to look at the contribution of skeletal muscle and adipose tissue. So skeletal muscles differing in type 1 fibers, and adipose tissue, sub-Q and visceral adipose tissues. And then here's just some examples of some of the PET image analysis that Heather and Julie and her group have done. Here's the vastus lateralis. We also are looking at biceps femoris, which has a higher type 1 content. And then sub-Q fat, we were just drawing circles. Now we're segmenting, Heather's segmenting all of the sub-Q and then matching it to the PET. And then here's our latest visceral and subcutaneous in the abdominal region. And so these whole images, regions are being mapped on the PET data. So we're not just looking at a little sliver like has been done in the past. And part of the problem in lean women is sometimes you don't have enough visceral fat to examine. So we're hoping that this overcomes that. So my first competitive revision for the first study was an intervention study in obese women. And it got a good score, but not quite good enough. And so in response to that, my program officer called me one day and asked me if I was interested in applying for an R56, which I'd never heard of before. And so I did, and it was successful. And so that's resulted in a study of insulin sensitivity mitochondrial function in women with obesity. And this is affectionately known as the MISS study. And so we enrolled 10 African-American and 10 Caucasian women with obesity. They were fairly well matched for age, BMI, weight, fat-free mass, again, tended to be a little higher. Body fat tended to be a little lower, but in this case, it wasn't significantly lower. And also not different was trunk fat or waist circumference, which isn't on here. It tended to be lower, but it wasn't different. Hemoglobin A1C was the same between the two groups. So we didn't have impaired glucose tolerance, supposedly. So this is sort of a busy slide. I've got the data from both studies, the lean African-American and Caucasian women, obese African-American Caucasian women at all the time points during the clamp. So this is hepatic insulin sensitivity was about 22% lower in women with obesity. So, but there was no racial difference. And so if we focus on the steady state 1 or 120 minutes, you can see we're up here at about 66% inhibition in the lean, and we're down to less than 60 to 50 to 55 to 60, 20% lower hepatic insulin sensitivity with obesity, but no racial difference. Peripheral insulin sensitivity was dramatically lower in the women with obesity, 60% lower. You don't need any statistics to see that going from up here to down here. They were really much more insulin resistant than the lean individuals, so nothing earth-shattering there. But when I look at just the obese, I don't see a significant difference in insulin sensitivity like I had anticipated. But I think part of the problem was a power issue. So if you look here, the 30-minute was actually 33% lower. The 60-minute was 30% lower. And steady state 1 was 16% lower. And no difference at zero at steady state 2. It just isn't there. And this gets back at this data from that earlier slide. And one reason I wasn't really concerned about these two studies showing no difference is because of the whopping insulin infusion rate they used. And the reason I say that is when David Kelly was still at Pitt, when we had our lab meetings, he was presenting some of the PET imaging data in obese and diabetic individuals. And I don't think it's been published yet, but he showed that the reduced glucose uptake that is seen in obese and diabetics was overcome with higher dose of insulin. So the defect is overcome with a high enough insulin level. So I think it was 120. So I wasn't concerned about these two initially, and I had powered it on these kind of differences. So 10 per group was plenty to detect this. So I still think, and I think maybe this should be followed up on, is that at least in obese African American women, there is a racial difference in glucose uptake, but it's overcome with higher insulin. So what that means, I don't know, but we didn't find a racial difference in the obese women. But we did see a significantly lower mitochondrial respiration, just like with the lower glucose uptake. So if you look at the blue bars versus the green bars, you can see dramatic reduction in, lower state 3 and state U respiration compared to the lean group. But the racial difference is still maintained. And then when we include both the Metflex, the lean, and the obese participants, the relationship between insulin sensitivity and mitochondrial respiration is stronger. Of course, you have a wider range, but it's a pretty strong relationship. So I was also fortunate to hook up with Shruti Shiva at Pitt, and she does a lot of platelet mitochondrial respiration. And she agreed to work with me on this project and do the platelet mitochondrial respiration. And there was a trend for lower basal, and except for this one outlier, which we'll discuss later, there was a racial difference in platelet mitochondrial respiration. And the relationship between platelet and muscle respiration was pretty strong, R.676. And one reason we were kind of interested in this is perhaps with a longitudinal study, we could look at changes in mitochondrial respiration in plasma, in blood samples, as opposed to doing all those muscle biopsies. So I think they, Shruti and Brett, published this, more data on that and showing that, and I know somebody else did too. Shruti also did mitosox, the ROS production in skeletal muscle and platelets, and did find increased ROS production in skeletal muscle and platelets. And then I did some measured protein carbonyls in some skeletal muscle homogenates, and I found a significant difference, but it's pretty noisy data. I wouldn't hang my head on that, but it follows the hypothesis. But I don't know how much faith I put in that. So in women with obesity, the hepatic and peripheral insulin sensitivity were significantly lower. There was no difference in hepatic insulin sensitivity. And although there was no significant racial difference in peripheral insulin sensitivity, there was a trend to be lower during the first phase of the plan. And mitochondrial ROS production was higher in the African American women. So that's the insulin sensitivity. We've also been looking at the role of mitochondrial respiration in lower RMR and VO2 max in African American women. And so RMR has been shown to be lower in African American women in many studies. And here's our data from our RENEW study, an intervention study in severely obese women, significantly lower RMR in the African American women. Here's the Metflex and MIST data combined, the lean and obese, significantly lower RMR. in the African-American women. And no one's really understands why that is. And so here's the one thing I wanted to point out that I found from our data here is, I think you all know that when you're normalizing RMR, you can't divide by fat-free mass because there's a non-zero y-intercept. The same thing occurs for mitochondrial respiration versus mitochondrial content. So definitely non-zero y-intercept, and the relationships between respiration and mitochondrial content are highly significant. The African Americans having lower content and much lower mitochondrial respiration. So most people just divide, mitochondrial respiration by mitochondrial content, usually mitochondrial DNA, which is also a messy measure. There's problems with that as well. So I've not seen that before. So mitochondrial respiration was highly associated with VO2 max, and that's been known for years. And then I don't know that I've ever seen a relationship between mitochondrial respiration and RMR, but we did find a significant relationship there. And when we included mitochondrial respiration in models for VO2 max and RMR, it eliminated the significant racial effect, suggesting that mitochondrial content plays an important role, respiration plays an important role in these racial differences. So with all of these findings we're finding with mitochondrial function and racial differences in RMR, VO2 max, insulin sensitivity, led us to explore the role of mitochondrial genetics. And I think you're all aware the mitochondria has their own genome, and it's unusual. It's a circular double-stranded DNA, and it's relatively small, only 16,500 base pairs. So it's easy to get a full sequence of the mitochondrial genome at high repeats. And one of the major features of mitochondrial DNA across the population is mitochondrial DNA haplotypes. And these are defined by a set of paternally inherited ancestral polymorphisms, starting from the L haplogroup, which is considered the root of the human mitochondrial DNA haplogene tree. And so you can see in Africa, the L, 1, 2, and 3, Y0 is not there. And then you see all these other haplotypes all across the world. And so, unfortunately, I wasn't familiar, really, mitochondrial genetics wasn't on my radar when I did the first study. It wasn't until a paper published by Greg Tranoff from the Health ABC study, who I didn't know at the time, but who I'm now collaborating with, showed this data, and this really piqued my interest in mitochondrial haplogroups. So resting metabolic rate in those with L2, 3, and 0 had significantly lower RMR than the typical European haplogroups, and L1s was sort of intermediate. And then about the same time, Todd Benini, who I also knew because we submitted a grant together, which didn't get funded, showed a strong European ancestry in African Americans was strongly associated with higher RMR. So I was fortunately familiar with those studies, so I included mitochondrial DNA and ancestry analysis in the MIST application, the study in obese women, but it's a small study. But we were fortunate to have in the freezer some Buffy coats from the RENEW study, which gave us a more robust sample size. We had 40 African American and Caucasian women combined. And I worked with the University of Pittsburgh Genomics Corps for the mitochondrial DNA sequencing analysis, but they had no idea how to analyze the data. I was able to find a program online called Mitochondrial DNA Server to analyze the mitochondrial DNA results, but since I didn't know what I was doing, I contacted a colleague at Pitt, Brett Kaufman, who's a mitochondrial DNA guy, and he told me that the program works well. It puts all those pieces together and gets to the right mitochondrial DNA sequence and identified the SNPs properly. So I had more confidence in the data that I was putting out. I was also fortunate that that at Pitt there was an investigator, DeLany, who had developed a panel of ancestry informative markers for estimating African and European ancestry and mixture. She had the primers, extra primers stored at the genomic score, and she was willing to collaborate with me. So, because I had no idea how to do any of that, so I was fortunate with that. So, repeating Todd Manini's paper, we did find that increasing European ancestry, increasing tertiles from 0 to 33, 34 to 66, or 67 plus, was associated with higher RMR, and those in the highest tertile of European ancestry had a significantly higher RMR than the first. We also found that repeated Greg's data showing that the L haplotype was associated with lower RMR than European haplotypes. And also, we had 3 African-American women who had the same percentage of African ancestry, but had European mitochondrial haplogroups. And in those three women, they had an RMR that was not different from the Caucasians. In fact, was not different at all. And that they were actually higher than the 19 who had African mitochondrial haplotype, an L of some sort. And so when I looked to determine which was the most important, I put race in the model, and then I added ancestry. It selected ancestry over race, and we have a higher R squared, higher percentage of variance explained. When I then added haplogroup, just L versus European, the haplogroup end of the model, as opposed to race or ancestry, and the variance was increased, that was explained. And then when I included L and 7 European haplogroups, you see another substantial increase in the R squared. So haplogroup seems to play a strong role in RMR, not only in the racial difference, but within the Caucasian group as well. So, okay, so unfortunately, I didn't have VO2 max in the Renew study. I only had it in the MIST study, so we only have 10 subjects here. But VO2 max was lower with the L haplogroup, and I only had 1 African American here that had a European haplogroup, tended to be higher, and this model explained more than race alone. And furthermore, mitochondrial haplogroup is in 2 very recent studies have been shown to play a role in the VO2 max response to exercise training. In one small study from the Heritage study, and then in a larger study in Finnish military conscripts. And so, mitochondrial haplogroup seems to play an important role in VO2 max and VO2 max response. And we had platelet and skeletal muscle mitochondrial respiration, but only in the MIST study, not in the Renew. So we have smaller sample sizes. And so we have this one outlier, so it was really funny how this came about, how we figured this out. So we were having a School of Medicine research faculty mixer one evening on top of some building at Pitt, and Shruti brought this data over. She goes, look, we have a lower respiration in platelets like you see in muscle, except I have this one outlier. And I had just finishing up the mitochondrial DNA analysis, and I remembered that we had 1 African American that had a European haplogroup. And sure enough, it turns out it was that one individual. So that was kind of exciting when you come across something like that. At a mixer, so we're sitting having drinks and going over data. That was fun. And we found the same relationship in skeletal muscle. So a much lower respiration in the L haplogroup. So, and then there's been not recent, but subsequent papers showing that mitochondrial haplogroup may also play an independent role in insulin resistance. particularly in an Asian population with the B4 haplogroup. And then they've done some hybrid cell studies with same nuclear, but with mitochondrial DNA with different haplogroups. And that showed differences in LUT4 translocation, AKT phosphorylation, and higher ROS production with the B4 haplogroup. So it looks like the mitochondrial haplogroup plays a role in insulin resistance as well. So I continued my mitochondrial DNA work when I got to TRI, and I work with, we are fortunate we have a pretty good biorepository from a lot of studies there, and they collected Buffy coats, and they have myoblasts we're going to get into, but not yet. And so one study was from Dr. Goodpaster's Mirage study. I worked with Paul Cohen, and he knew Greg Tranoff from another study. So we hooked up with Greg for our mitochondrial DNA analysis guy. And we looked at the Mirage study we had a 4-meter walk time with increasing age. And it's not a lot of subjects. It's 6 African-Americans and 8 Caucasians. But there's a trend for increased time to walk 4 meters in the African-Americans compared to the Caucasians. And furthermore, we had 2 African-Americans in that group that had European haplotype, and they had a similar time to complete the 4-meter as the Caucasians. And if anything, this one was the quickest. So it was an African-American with European mitochondrial haplogroup. And so we use this data and a lot of the other stuff that I just presented, and a lot of preliminary data from Greg's studies and Paul's studies, and a submission for an ancillary study to the SOMA study. And our resubmission is being reviewed in 2 weeks. So hopefully we'll get good news on that. And then Brett and Paul and I are working on an ancillary to the motor pack to add mitochondrial DNA analysis. So in conclusion, lower mitochondrial capacity appears to play a role in lower RMR and VO2 max in African-American women. It may be related to increased risk for weight gain in African-American women, and a mitochondrial genetics appears to play a role in lower mitochondrial function, metabolic rate, and VO2 max. So the overall summary today is that African-American women have a characteristically distinct form of lower insulin sensitivity linked to abnormalities in skeletal muscle, independent of adiposity or physical activity level. This metabolic phenotype is associated with lower skeletal muscle mitochondrial capacity. The lower RMR and VO2 max in African-American women is associated with lower skeletal muscle mitochondrial capacity and appears to be related to mitochondrial DNA differences opposed to genomic differences. And whether the insulin sensitivity in African-Americans is associated with mitochondrial DNA is not known. I have a little bit of data from the MISS study, but it's a small study and it's a little too small to really get anything definitive out of that. And so, in summary, our findings linking mitochondrial capacity in muscle to lower insulin sensitivity, low RMR, and low VO2 max suggest to us that aerobic exercise training may be uniquely beneficial to African-American women and thus help personalized treatment of insulin resistance and obesity. And so, again, that was what I tried to do with the competitive revision, but maybe now we can get that funded. And so a lot of people helped me with all this at Advent Health, Brett and Paul, and Heather Cornell at our imaging center is outstanding, and Lauren Sparks has added adipose tissue biopsies now to the PET studies. And we have a wonderful core system here at TRI, and so they do an awesome job. And then Julie at Mass General. I didn't really talk at all about the Samoa study, but Steve McGarvey is the MPI on that, and Nikki Holly at Yale is just awesome at setting up the Samoa operation, and Greg Tranoff at California Pacific Medical Center. And then there was a whole bunch of people at Pitt, including starting with David Kelly. I mean, I went to Pitt to work with David Kelly on on the cool stuff he was doing with mitochondrial function and PET imaging and clamps, and then he left, went to industry, but I continued and learned everything I wanted to get from him anyway. So, and then Fred and Brett were big collaborators, and Paul and John Dubé. I'm still working with John Dubé. He's finishing up writing that fatty acid oxidation paper from that first study. And then funding NIDDK, NHLBI, and Evan Health. So that's my presentation.
All right.
Mute. Let's unmute.
Yeah, unmute and let's clap for our speaker. Thank
Oh,
you so much. That
unbelievable.
was so interesting.
So are there any questions?
Yes. So there are some questions in the chat. I don't know, Jopna, if you want to read those, or do you want me to read those, or how you want to navigate that? So Jopna started with a question: are there validated equations that account for these racial differences in our RMR and in energy requirement calculations?
I've got a paper showing that has racial has race added to it. I think there are other ones too, but from the Renew study, I had developed an equation that includes race to calculate the difference. And you know, another thing that I should have included here is, you know, so the African American women have seeing the energy requirement comment made me remember that I looked in the Renew study. We had intake balance by longitudinal doubly labeled water and DEXA measurements. And so I was able to actually calculate the energy intake during the calorie restriction. And the African American women and Caucasian women achieved almost exactly the same on average intake during the intervention. But they lost less weight, which is often seen as they lose less weight during intervention. It wasn't because they were less compliant. They were as compliant as the Caucasian women in both physical activity and intake. It's just that they were prescribed a lower deficit because they had lower energy requirements. They had lower RMR, which led to lower total energy expenditure and lower energy requirements. So that reminded me of that.
Thank you. Dr. DeLany, could you stop sharing your screen?
Oh,
So
stop sharing? Okay.
Yeah. Then we'll just read out the question.
Okay.
Thank you. All right, I think Dusty has asked, when RMR is associated with European ancestry, do you think the climate of the region may play a role, since cold weather normally induces thermogenesis and a higher metabolic rate? May this be why mitochondrial DNA is associated with RMR?
So that is an interesting and insightful question. So that part of the theory of the mitochondrial DNA tree is that as people move to colder climates, that their mitochondria changed to be, you know, less efficient and produce heat. And so there's some data to support it. It's not entirely clear that that's the case, but it's a very interesting and cool theory.
So I'm just going to jump in here because I think it's really fascinating. And I was interested in the fact that you saw UCP2 come up, which is an uncoupling protein in your skeletal muscle. So what do you make of that? Any thoughts on why there's that relationship?
Yeah. You know, it could be energy wasting, less energy wasting, lower mitochondrial respiration. I mean, as I mentioned before, that we did some reading into all those differences that popped up, but it's a very complicated thing to figure out. But UCP2 was one that I agree that popped out to me too right away. Boom, UCP, that kind of makes sense.
Yeah, so we're real interested in uncoupling proteins in white fat. So we certainly see relationships between UCP1 and insulin sensitivity in white fat and brown fat. And so my next question was going to be, are you doing anything with that? Are you planning on doing similar analyses with the mitochondria from that biopsies?
To be honest with you, I don't know what Lauren Sparks is in charge of all that. I know she's trying to grow up some cells, and she's doing some things in the actual body. To be honest with you, I'm not sure what she's doing.
Well, lots of.
I don't, but I'm pretty sure that like they're not doing mitochondrial respiration in the adipose tissue biopsies. I don't think they're focused on mitochondria. But that's a good question. I mean, I'd be interested in that as well.
Yeah. Japna, do you want to read your question?
Well, okay, sure. So what I was thinking was, how do you propose incorporating this mitochondrial haplotype information, in terms of stratifying participants at baseline in studies that are investigating racial differences in metabolic outcomes? So how would you go about doing that?
Well, that would be an interesting approach. I don't know that maybe you wouldn't want to do it for for matching, but maybe as an outcome predictor, predictor. So and I haven't looked yet at the Renew data. So I have intervention data. I haven't looked at that to see the effect of mitochondrial haplotype on weight loss. But I've been meaning to do that. I just haven't gotten around to it. So because we do have a fair number. Plus I have more. I really need more data. I mean, I've got a lot of this mitochondrial DNA data. I don't have quite enough for a paper, but I've got more Buffy codes from the Renew study. I was able to bring them with me. And then there's some other biorepository samples. We'll have it for the MatPet study. We'll have more data from that. We're doing the mitochondrial DNA analysis and ancestry analysis, but by another method. So yeah, I don't know that we're quite there yet to do that.
Okay, thank you. Looking forward to seeing what you find. Especially as you're moving towards precision nutrition, these are the kinds of questions people are most interested in. Like, well, how do you do with all that information?
Yeah.
Jim, can I ask this is a lot of data for me, and I'm trying to get a gross generalization so I can at least put it under an umbrella. And it seems like African Americans have insulin resistance or high risk of diabetes or whatever. And that is somehow a pancreas over secretion of insulin and a muscle problem, that axis.
Right.
Whereas Hispanics, their high blood glucose seems to be related to fatty liver and overproduction of glucose by the liver. Is that sound at all make sense? Or
yeah, it does. Yeah. So
because I don't think HGP correlates with blood glucose in African Americans as well as it does in Hispanics. That's what I'm thinking.
And the other thing that I'm interested in looking at in the PET study is I did not appreciate how much glucose uptake occurs in adipose tissue.
Yeah. Vicky's got data.
Because when I started all this, I said, well, peripheral glucose uptake, that's skeletal muscle. But Brett and David did a study with looking at adipose tissue uptake, and in women, particularly, not men, it was like 14% of glucose uptake occurred in adipose tissue.
And particularly if you have a lot of adipose tissue, then it accounts for this huge amount of RD. Yeah. Wow. I think Jill has a question in here. She said, I'm not sure if you mentioned this at the beginning. Is the fiber type similar between African Americans and Caucasians? And I think it's not, right?
No, it's not. So, it's not remarkable, but type 1 fibers were lower in African Americans, and type 2 was higher. And, there wasn't a whole lot of data when I started, but, interestingly, some of the African sprinters, some of the best runners in the world have lower type 1 and higher type 2 fibers. So it seems to be a racial difference, lower type 1 and higher type 2.
Yeah. Well, Japna, we're over time.
I think okay, one last question, and then we'll let you go, Dr. DeLany. So Talia has a question. She's saying there's a growing prevalence of multiracial, multi-ethnic individuals in the US. So given the data presented about mitochondrial haplotypes and the apparent improvement in mitochondrial respiration when African-American women have a European-derived haplotype, do you anticipate that the future US population will have better or worse insulin sensitivity and diabetes risk?
So, well, I hadn't thought about that at that level. Since I don't really know if the insulin sensitivity is different by mitochondrial haplotype. I mean, it could potentially, but there could be other ramifications. Having, maybe it's in some things it might be good to have European haplogroup, but others, there might be a disconnect between the genomic, because most of the mitochondrial proteins come from the genome, and so there's got to be a proper interaction between the mitochondrial and genomic protein. So that's a complicated question. It's a good one, though.
That was a great question, Talia. All right, well, thank you very much, Dr. DeLany, for still presenting the seminar despite what you've gone through in the past 24 hours.
Well, thank you. Thank you for inviting me. I hope you did.
Thank you very much.
Wonderful.
Let's give him a round of applause.
Okay. Thank you all.
Thanks so much.
Thank you.
Okay.
Yeah, great.